Hello and welcome to this video on insulin derived from yeast. This is the modern insulin product that many diabetics would be familiar with. It is sold under a number of brand names such as Humalog and NovaRapid. Insulin that we use today is a product of genetically modified yeast and E. coli, with yeast the preferred microorganism. It is a homologue of the normal insulin peptide. This has been refined, purified, and packed for use in humans. As most people with a passing knowledge of diabetes will know, insulin is needed for the body to respond to blood glucose levels appropriately. Cells have something called an insulin receptor on the surface. The most prolific expressor of these receptors are skeletal muscle and hepatic cells. By mass, it is the skeletal muscles. Insulin binds to these receptors, and this activates a mechanism that brings more glucose channels or gates to the cell surface. This effectively increases the potassium uptake to support the channels, which increases the cell's energy stores. In the liver, these are converted to fatty acids, glycerol, and then stored in white adipose tissue, which is better known as fat. Diabetic patients have a problem with these receptors, where they stop activating that cascade of events, culminating in glucose channels. This reduces how effectively the body responds to blood sugar levels. The exact pathogenesis of type 2 diabetes is unknown, but involves damage to the cells that express insulin, although more commonly found in type 1 diabetes. The insulin itself can be targeted by immune cells, or the cell receptor is defective. This generally leads to a gross undersupply or ineffective use of insulin. Modern technology has helped us to overcome this and create further variations on insulin. The important two being fast-acting and slow-release insulin doses. In the next 20 years, the World Health Organization has estimated that insulin's value as an industry would go from $12 billion to $54 billion globally. As insulin is a molecule, it is quirky and interesting. It is a peptide. That means that it is like protein chains and even messenger RNA. A translation of the gene that codes for insulin is just a long string of amino acids. A long string of interconnected units. To keep this structure called pre-proinsulin stable, it is shaped like an E. A shape coiled around itself. As it is passed out of the pancreas, cells responsible for production of the pre-proinsulin signal is clipped off, which just leaves the proinsulin molecule. The proinsulin molecule has three core parts, which are called domains: an amino terminal B chain, a carboxyl terminal A chain, and an interconnecting C peptide. It is the C peptide which is first removed from the insulin. This leaves behind the alpha and beta domains via weaker inter-unit connections, but this also modifies its shape. This allows for specific binding to the insulin receptor, going as far back as ancient Egypt. The ailment we know as diabetes has been around, with reports of sweet urine due to the excess sugar that is found in the blood. It was not until the early 20th century that we found a way to counteract this disease through artificial supplies of insulin. This was not human insulin, which is what we effectively have now, but sourced from bovine or porcine animals, these being oxes and pigs. This early, crude development was a combination of animal pancreas juices mixed with a carrier, that was then injected into a 14-year-old diabetic boy in Toronto, Canada. He reacted both very well and very badly to this early insulin product. He was able to show a slight improvement from the insulin that he received. The same insulin shot was so contaminated and crude that he also had an allergic reaction. This adverse event led to future tests being cancelled. If only a cure was so easy to make. Over the next 12 days, the main researcher behind this, James Coli, worked continuously to improve the pancreas juice extract that he had made. This led to a second dose that was given a short time later. This was, relatively speaking, a complete success. There were no obvious side effects, the immunological ones being important, but also resulted in complete reduction of glycosuria, which is that excess blood sugar levels. Woohoo! A successful cure. But no. In the years that followed this, there was work towards creating an effective and mass-produced insulin product. This was then sold off to Eli Lilly, a major pharmaceutical company. In order to produce 8 ounces of insulin, they needed more than 2 tons of pancreases from pigs. An average pig pancreas is roughly 350 grams. 2 tons would therefore require just under 6,000 pigs. In 1922, a time in which the USA consumed roughly 15,000 pigs a year, this was a huge problem. Each unit of insulin is approximately 0.0347 micrograms. 8 ounces is 227 grams, and 1,000 micrograms is a gram. With a little bit of math, we can conclude that just the one 8-ounce dose of insulin would provide for 6,541,786 units. In a modern setting, this would be 6 units per diabetic person who consumed 60 grams of a carbohydrate-rich food. This gives a little over 1,090,297 doses. The exact number needed could be slightly higher or lower. Either way, it is only enough insulin to give 2,987 people 1 dose each day of the year. With more supply or better management, that figure could rise, but there was always going to be a major limitation in the supply chain. How many pigs and cattle could be raised, and how much time they took to rear. A fact recognized very early on in development of the human autologous insulin. This long-winded story leads up to the development of autologous or synthesized insulin, which is a modern miracle of pharmacology. It took over 56 years to reach this point. In 1978, a recombinant DNA technique was used to put the insulin gene into E. coli, which was the first major breakthrough in the development of modern insulin. E. coli was, and still is, a model organism for research. It was well studied, expanded at a geometric rate every 20 to 30 minutes, and was relatively easily modified. Think how effective you could be if your product doubled in size every 20 to 30 minutes. This was the thinking behind the placement of genes that encoded the human insulin and the human growth hormone into E. coli between 1978 and 1979. This is where one of the bigger problems with using another organism to produce products comes in. The various post-translational modifications that occur in the messenger RNA and final product were not able to be conducted in the E. coli. There were problems with glycosylation, phosphorylation, processing, and formation of certain bonds within it. These were essential for the insulin to be biologically active. Since they did not occur in E. coli, the insulin produced at this stage could not be used as effectively. Since it was first produced in E. coli in 1978, the company behind that, called GeneTech, developed a technique that allowed them to create both the cDNA for the A chain and B chain. This was then put into the E. coli as two separate units. These would then be expressed, and once expressed, the A chain and B chain could be extracted from the media the E. coli was growing in. This gave them the two main and important parts of the insulin product. They could then be purified and incubated in a situation in which they could then cross-link. This gave them intact and, most importantly, bioactive insulin. It was a more difficult, time-consuming, and error-prone process, but it meant that they could produce insulin. There was another problem, particularly with the use of E. coli, and that regards codons. Codons are the three nucleotides which correspond generally to an amino acid. By using one of the possible three matching codons, you can then select for a specific amino acid. Since there are so many codons that could possibly occur, the number of amino acids is smaller, which means there are redundancies. Some species rely on particular codon sequences over others. E. coli is an example of this. The usage of certain codons in E. coli meant that trying to insert a gene that was not consistent with the species led to a reduced expression level in that species. By taking codon usages in E. coli that were heterologous, they were capable of changing the gene just slightly. This then allowed for the E. coli as a host to express human insulin. Another option was, rather than try and create two separate chains as was occurring in 1978, it was possible through the use of a synthesized complementary DNA strand to put the entire gene along with a promoter into E. coli. This created proinsulin, the step after that promoter tail had been taken off. This allowed for both the creation of a full insulin molecule with the C-peptide taken off. This was more efficient and convenient in production of insulin, as it didn't require the two chains be combined separately. It has been the dominant method used since 1986. These first-generation techniques all were identical to the human insulin and were better than the animal-derived insulin products. This is essential, as insulin is not meant to cause an allergic reaction. As with blood transfusions, a repeated stimulus of the immune system by synthetic insulin could lead to an ongoing immunological reaction to both innate and prescribed insulin. This would gradually kill an individual in a slow, sure, and gruesome manner. The difference in pork-derived insulin is just one amino acid. In bovine insulin, it was 3, but this is still enough for the human body to recognize a foreign element and aim to destroy it, and likely cause considerable collateral damage in the process. Since 1986, there have been major advances in the fields of genetic engineering and the technology that goes with this. It has allowed researchers and pharmaceutical industry to chemically synthesize genes that have altered nucleotide sequences. This is important if you wish to alter the insulin product. Alterations in insulin are predominantly done using the amino acid sequence that it is made from. It's been noted over the many decades of research into insulin that there is usually some sort of oligomeric form. Oligomeric means that there's differences in what keeps the alpha and beta chain together. Sometimes it can be zinc-containing hexamers, sometimes disulfides, and a few other variations. This is very important when you are looking at changing its biological activity. If you can make it so the oligomeric complex breaks down in different ways depending on what it's consisting of, you can alter the effectiveness it has. Think about what would happen if you could speed up or slow down the effect of insulin injections. And that's exactly where researchers went to. The original design behind injectable insulin was that it would be a subcutaneous injection. This would gradually leach into the blood. The initial injection would take 2 hours to reach a peak, and that would be sustained for 6 to 8 hours. Long-term stability, so long as you did not consume any large amounts of carbohydrates in that 6 to 8 hour window. This presents a significant problem. How do you deal with those spikes in glucose that occur after consuming food? And it led to the development of fast-acting insulin. In order to create this product, some modifications had to be made to the amino acids. Primarily, they were able to look at changing only a few very subtle but critical parts of it. These were primarily in the B chain. This allowed them to create an oligomer of the original insulin. Fortunately, they were able to do so in a way that would not instigate an immunological reaction. Commercial production is exemplified in the Lispro. This is a form of synthetic, fast-acting insulin produced by changing out the lysine at beta chain position 28 for proline at position 29, a small swap, but made it effectively able to work that much quicker. There are others that have been developed since that have changed other alternative amino acid residues within that. This has allowed for the introduction of more and new insulin that is fast-acting. So far, all of this is applicable to both yeast and E. coli. The reason yeast is preferred over E. coli owes to a fundamental difference in how they function. In many respects, yeast is superior as it is capable of conducting the necessary post-translational modifications. This means that a plant manufacturing insulin does not need to go around adding in extra steps to modify the insulin. It can all be done within the yeast cell and still be biologically active. Many of these include phosphorylation, glycosylation, acetylation, and acylation. Important steps in the development of a biologically active insulin for efficient expression and secretion of recombinant proinsulin in yeast. The insulin gene had to be engineered to contain both the alpha chain and beta chain, but not that C peptide. This is why that ability to have post-translational modification is important. Normally, that C peptide is removed down the track from the original expression to keep it stable. By creating a slightly modified gene and using the systems that are within the yeast cell, pharmaceutical companies can do away with the step that requires removing the C peptide entirely. This involves taking a DNA sequence that already fuses the A chain and the B chain with an internal signal in the yeast cell itself. This means they can get up to 80 micrograms per milliliter of insulin. This method of insulin production is capable of producing 80 grams of insulin per liter. That would require roughly 28,375 liters for the same production of insulin as was obtained in 1922. In bulk, this number could be achieved with something similar to a microbrewery. Admittedly, it would require a lot of microbreweries, but still, viable on a small-scale production. In fact, one of the 3 major manufacturers of insulin, Eli Lilly, achieves their production using 5,000 vats of 50,000 liters each in a roughly 420 kilometer facility. At present, there is considerable unrest over the cost of insulin. As you can see, this is based largely on the difficulty in getting the drugs made in the first place. It required highly specialized tech that only became available in the last 40 years. This, in combination with the previously described costs and difficulty in getting novel pharmaceuticals approved, is a significant part of this growing cost for insulin. The final part is recovering costs before patents expire. This will be a less significant concern, as there will be more patents expiring over the next few years than many of these companies are yet prepared for. As stated early on in this video, insulin is a modern miracle of pharmacology that it has taken us nearly 100 years to get to a point where we have at least a viable, ongoing solution to both the acute problems of diabetes and the chronic ones. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.